Good morning, students. Welcome to the class. In last class we have discussed about some of the genetic diseases. So now let us discuss about some more genetic disorders, particularly gene disorders. Now let us move to thalassemia. Thalassemia is genetic disease. It is a autosomal linked recessive gene disorder or blood disorder. So you can understand the terms here. Autosomal linked recessive blood disorder. Autosomal linked recessive blood disorder, and it is transmitted from unaffected carrier parents to their offsprings. It is transmitted from unaffected carrier parents to their offsprings. Okay, so now what are you understanding by this term? Okay, autosomal linked recessive blood disorder. So now the thing is, you have to understand here, the disease is mainly caused because of the mutation of the gene. It is caused due to the mutation of the gene, or it is a deletion of a mutation or deletion of a genes, loss of a gene, deletion of genes. Because of this loss or deletion or mutation of a genes, a defective hemoglobin is produced. Okay, there is a defective hemoglobin, or reduced rate of synthesis of hemoglobin takes place. Reduced rate of synthesis of hemoglobin takes place. Reduced rate of synthesis of hemoglobin takes place. Okay, or globin chain takes place. So now here you have to understand certain important idea which are present in this. So now thalassemia, you have already studied sickle cell anemia. In case of sickle cell anemia, you know that very well. Hemoglobin, first of all you should know about the hemoglobin. Hemoglobin is a protein, blood protein, which is made up of two chains. One is called as the alpha chain and another one is called as the beta chain. So hemoglobin is a protein which is made up of two chains, and both the chains are linked together by disulfide bridges. So now this makes one complete hemoglobin molecule, and which is made up of a different number of amino acids. So each chain is made up of a different amino acids are present, amino acid residues are present here. Okay. So in this way, this makes a normal hemoglobin molecule. Now what happens here in case of sickle cell anemia? Now I am talking about the sickle cell anemia. So you have to understand about the difference between the sickle cell anemia, okay, and thalassemia. In case of sickle cell anemia, now just understand here, in case of beta chain, so this is sixth amino acid must be glutamic acid. So instead of this glutamic acid is replaced by valine. That is because of the defective gene. Okay, presence of a defective gene. So in this way, what happens here, in case of sickle cell anemia, the quantity of the protein remains the same. Okay, the length of both the chains, alpha chain and beta chain, remains the same. But if you think of the thalassemia, just you can compare. So now this is thalassemia. So here this is alpha chain and this is beta chain. So here the length, you can just observe length of the globin chains. So this alpha chain is smaller than this beta chain. It means what has happened here, okay, there is a reduced rate of synthesis of one of the chains of this hemoglobin takes place here. Okay, so this is because of the gene, defective gene, the defective gene which is transmitted, which is carried from unaffected carrier parents. Unaffected parents, it is transmitted. So in this way, now question, you may ask a question, whether this disease affects only to the alpha chain or it also affects even to the beta chain. So like that you may pose a question to me. So now I can tell you, so this gene, so here since both the chains are present and it may affect either to alpha chain or to the beta chain. Are you following me? Okay. So now based on that we can classify thalassemia into two different types. Types of thalassemia. Types of thalassemia. Thalassemia are mainly classified into two different types. One is called as alpha thalassemia. Alpha thalassemia. Alpha thalassemia, and another one is called as beta thalassemia, can be represented like this, beta thalassemia. Beta thalassemia. Why these are the different types? There are different types. Just now I told you here in hemoglobin which chain is mainly affected, whether it is alpha chain or beta chain. Based on this we can classify thalassemia into two different types. If mainly alpha chain is affected, then the thalassemia is called as alpha thalassemia. If reduced rate of synthesis of beta chain takes place, then it is nothing but a beta thalassemia. Now one more thing is, you may ask a question, what is the reason for, what is the cause for this alpha thalassemia? Alpha thalassemia is because of the defective gene which is located on chromosome number 16 of both the parents. In case of alpha thalassemia, defective gene is located on chromosome number 16 of both the parents. Two closely linked genes present on chromosome number 16. And beta thalassemia is caused due to single gene present on chromosome number 11 of both the parents. So it means what we can understand here, whether it affects alpha chain of the hemoglobin or beta chain of the hemoglobin, that depends upon the defective gene where it is present. Okay, if defective gene present on the chromosome number 16, then what happens? It causes alpha thalassemia, and defective gene is transmitted from chromosome number 11 of both the parents, then it causes beta thalassemia. Okay, here two closely linked genes which are transmitted from both the parents to the children. Here single gene present on chromosome number 11, it is transmitted from parents to offspring. In this way, we can explain this thalassemia. In case of thalassemia, just we can understand the relationship between or the difference between sickle cell anemia and another one is thalassemia. In case of sickle cell anemia, the length or quantity of hemoglobin remains the same in case of sickle cell anemia. But here in thalassemia, what is happening? The reduced rate of synthesis of globin chain takes place. So therefore, sickle cell anemia is a qualitative problem. It is a qualitative defect, qualitative disease, because your quality is reducing. Just one amino acid is replaced, glutamic acid is replaced with the valine. In case of a sickle cell anemia, therefore it is a quality, and it reduces the quality of the hemoglobin also, and it reduces the quality of transport of oxygen also. And in case of thalassemia, what is happening here? So it is reduces the synthesis of hemoglobin. Therefore, this is a quantitative problem. Quantitative defect. This is because quantity of hemoglobin is reduced here. So in this way, you can differentiate between. Okay, in the examination they may ask question, how thalassemia is different from sickle cell anemia. So thalassemia is a quantitative defect and sickle cell anemia is a qualitative defect. Okay, here why it is a quantitative defect? Because there is a reduced rate of synthesis of hemoglobin takes place, that is because of the defective gene. And sometimes examination they may ask a question, write the differences between different types of thalassemia, alpha thalassemia and beta thalassemia, and you can list out different types of diseases. And let us move to one more disease of this lesson. Okay, now let us see, that is phenylketonuria. Phenylketonuria. This disease we can call, in short we can write it as PKU. So in this way also you can write this disease. What is this phenylketonuria? How it is different from other diseases? Now look here, this disease can be defined as it is a error of metabolism. Error, I can tell you, error. Error. You can understand what is the meaning of error. Error of metabolism. Error of metabolism. Error of metabolism takes place sometimes during our lifetime also. Okay, sometimes suppose if you take a food, heavy meals, then digestion may not take place because of the less secretion of enzymes. That is the error. But this error is transmitted from parents. So therefore this is called as an inborn error. Inborn error of metabolism. This error has not occurred, okay, in that particular day or in the life of an individual, but it is transmitted from the parents. So therefore it is a inborn error of metabolism. Okay, inborn error of metabolism. And okay, so this kind of disease, this kind of error, what we call it is a phenylketonuria. What is happening in this disease? So generally what happens here in our body, that if particular gene, normal gene is present, enzyme is produced. So normal enzyme, that normal gene, that which is present on a particular location of the DNA, and it can produce a enzyme. What is that enzyme? Phenylalanine hydroxylase. Phenylalanine hydroxylase is the enzyme. Don't get confused, which is already mentioned. We have already explained regarding this enzyme. Phenylalanine hydroxylase enzyme for production of every enzyme. Okay, gene is necessary, which is present on the chromosome. What is different enzymes are performing different functions in our body. Now, this enzyme, what is the function of this enzyme? This enzyme is responsible for conversion of phenylalanine. Phenylalanine in our body, it is converted, it is metabolized into tyrosine. It is converted into tyrosine. This process, this conversion should take place. It has to carried out. Okay, in every person, in normal person, this conversion takes place, because in normal person, normal dominant gene is present. This dominant gene produces the enzyme. So this phenylalanine hydroxylase enzyme is produced. This enzyme is responsible for this conversion. Tyrosine takes place. Suppose if the individual do not have this gene, or defective gene is present, or recessive gene is present, now what will happen? And this enzyme will not be produced. This enzyme will not be produced. When the enzyme is not produced, in the lack of this enzyme, phenylalanine hydroxylase, this phenylalanine, it remains as it is, and it gets accumulated in the body, in the blood, and it causes different defects, which we have already explained. Okay, accumulation of phenylalanine and its derivatives, it leads to, okay, mental retardation. Mental retardation takes place. Mental retardation takes place, and excess of phenylalanine, which is accumulated, it gets excreted through urine, and when the urine comes outside and it reacts with atmospheric moisture or oxygen, and urine turns to black color. Therefore some people call it as a black urine disease also. Black urine disease. Okay, it is also called as black urine disease, since urine turns to black color when it is excreted out. And this kind of disease is called as a phenylketonuria, and it is one of the inborn error of metabolism. Inborn error, you can understand, inborn, therefore it is nothing but a genetic disease, and it will be transmitted from one generation to another generation. Okay, we can understand here, because of the defective gene, enzyme is not produced. When the enzyme is not produced, this chemical metabolism will not take place. Here you can understand. Okay, this metabolism does not take place, and because of a lack of conversion of this, and that gets accumulated, and it causes a mental retardation, and that excess of phenylalanine and its derivatives will excrete through urine, and when urine comes outside, it turns to black color. Therefore the disease is also called as a black urine disease. Okay, so now you can follow all these different types of diseases, and one more disease which we have listed already in the last class, that is cystic fibrosis. which is not explained in your textbook. Okay, so there defective gene is present in case of cystic fibrosis. Just little I can tell you. Okay, in case of cystic fibrosis, okay, that mucous membrane gets affected. Mucous membrane gets affected. That mucous membrane, particularly in the respiratory system, the mucous membrane is affected. Because of that, what happens? Person suffers from shortness of breath. That is what we call it. This is cystic fibrosis. And since it is not mentioned in the textbook, let's not go detail into the disease. Okay. Now let's move to one more important disease as per your syllabus, that is color blindness. Color blindness. Okay. Now what you can understand by seeing the term color blindness. You know that very well. In nature, different animals will have a different capacity, but particularly human being has a capacity to differentiate or identify various colors. I can tell you, human being is a unique animal or the only animal which has got capacity to identify different colors, differentiate different colors. How are we able to identify different colors? That is because of the genes. Okay, we have got a gene on our X chromosome. We have got a gene on our X chromosome, and these genes makes us to identify these different colors. So this gene must be present in the dominant condition. So gene responsible for identification of a color or differentiation of a color should be present in the dominant condition. Okay, and now, so this gene, it synthesizes a protein. Different proteins are synthesized, and what we can call them as a cones. I think you might have heard about the words cones and rods, which are present in our eyes. Okay, so this particular protein, when it is synthesized, different cones are synthesized, then we are able to identify different colors. Okay, suppose if this cones for particular color is not synthesized, then we are not able to see. We become unable to identify that particular color, and we become a color blind. So in this way, okay, so this color blindness was first identified, and this disorder was first noticed by a scientist known as Dalton. Therefore, this color blindness is also called as a Daltonism. And more common color blind is red-green color blindness. Red-green color blindness. Red-green color blindness is more common one. More common color blindness. Red-green color blindness. Red-green color blindness is a very common type of a color blindness. What is the reason for this red-green color blindness? That is because of the absence of a gene, or that is because of the recessive gene. Recessive gene has become recessive, which is present on the X chromosome. Okay, and when the gene becomes recessive, these particular proteins are not produced, which are responsible for the formation of these red cones and green cones, so that we are not able to identify red-green color blind, red and green color, and it causes a disease known as red-green color blindness, and this is a very common one. Now, what are you understanding here? It is also sex-linked disease like hemophilia. It is a sex-linked character. Identification of a color, differentiation of a color is a sex-linked character because your gene is located on the X chromosome or the sex chromosome. Okay, and it is also transmitted. You know that very well. Like hemophilia, disease is transmitted from parents to the offspring. Okay. So one more thing you can understand here, since this gene is located on the X chromosome, it follows a kind of inheritance is known as skip pattern or crisscross inheritance. One more term I can tell you, crisscross inheritance. Crisscross inheritance. Why it is? Okay, here female, okay, it is carried from unaffected carrier mother to the some of our male children like hemophilia. So it is transmitted from unaffected carrier mother carrier mother to some of her male progeny, or to some of her sons it is disease is transmitted. Why it is transmitted from carrier mother to some of her male progeny? Since gene is present only on the X chromosome. Okay, male will have a X and Y chromosome. Okay, male individual receives X chromosome from the mother. Therefore, male children will be affected. Not all male, some of the male children will be affected. And this is a very, very important concept from the point of CET. Okay, they will ask this, certain probabilities from this. Okay, here, if you think like this, female to become colorblind, she must have a defective gene on both the X chromosomes. To receive, here one X chromosome is received from the mother, another X chromosome is received from the father. Therefore, here female to become so this colorblind is very rare, like hemophilia. Okay, so now here 8% of the males are colorblind, and only 0.4% of the females are colorblind. So we can understand the difference here. Why this female, the percentage to become colorblind is very less, because female to become colorblind, at least her mother must be carrier and father should be colorblind, then only she becomes a colorblind. Therefore, the percentage of becoming colorblind in case of a female is very, very less. So, these are all different types of genetic disorders, gene disorders, particularly gene disorders or Mendelian disorders. So, these are all can follow the principle of Mendel. Therefore, they are called as Mendelian disorders. Okay. Students, let us move to another set of genetic disorders. They are called as chromosomal disorders. Which we have already discussed. What are these chromosomal disorders? So you know that very well. Chromosomes, the particular individual will have a definite number of chromosomes, particular set of chromosomes will be present. These disorders are mainly caused because of either increase or decrease in the number of chromosomes. One should have a fixed number of chromosomes. A species should have a fixed number of chromosomes. Either increase in the chromosome or decrease in the chromosome, which causes a disease known as chromosomal disorders. Okay, so here we have already discussed two different terms. One is called as aneuploidy and another one is called as polyploidy. I explained in the last class. So now let us discuss about some of the important disorders, chromosomal disorders. Now, just I can tell you what are the different chromosomal disorders as per your text or as per your syllabus. One is called as Down syndrome, and another one is called as Klinefelter syndrome, and next one is called as Turner syndrome. As per your syllabus, you have to understand. Now let's discuss about first one is Down syndrome. Down's syndrome. Okay, so now this Down syndrome, here you have two new words. One is Down, I think it may be new or it may be old for people. Down means down, but here that is not the meaning. And another one is called as syndrome. Here Down means it's not up and down, it is a name of the scientist who first noticed this disease. Okay, a physician who noticed the disease or who observed the disease. Langdon Down is the name of a scientist who first observed this disease. So therefore this disorder is called as a Down syndrome. Syndrome, I think you might have come across the word syndrome. Syndrome is not one disease, it is a collection of a disease, collection of disorders is called as a syndrome. Okay, for example, acquired immunodeficiency syndrome in case of AIDS. Okay, therefore the collection of diseases is known as a syndrome. Now you can understand here Down syndrome, Langdon Down, and here very important is you should understand about normal chromosomal complement. That is generally we have 44 chromosomes. These are called as autosomes. And along with autosomes, if it is a female individual, XX chromosomes are present. So therefore it becomes a 46. So this is normal chromosome of a female. Similarly, 44 autosomes plus XY is present, and which becomes a male individual. This is nothing but a normal chromosomal complement or karyotype. So you have to understand this is karyotype. In this way you can write it. Now for every disease you must know about the karyotype. Okay, here karyotype for this disease is, so I am writing karyotype here before this disease I am writing. Okay, now here 44 chromosomes. Here in this Down syndrome, and which affects only to the autosomes. So instead of 44, here 45 chromosomes are present. So normally 44 must be present, but here 45 chromosomes are present. What autosomes are 45? Plus X may be present or XY may be present, and which makes total 47. Chromosome number becomes 47. 45 here, one chromosome is increased. There is a gain of a chromosome. Okay, so you know that very well there are total 22 pairs of autosomes and one pair of sex chromosomes are present. And chromosome number, one particular chromosome is increased to the 21st set. Therefore, this disease is also called as a 21 trisomy. Okay, this disease is also called as 21 trisomy. Trisomy means because there is increase, there is a gain of a chromosome to the 21st set or 21st pair. Therefore, it is also called as a 21 trisomy. Now, what is the cause for this kind of chromosomal disorders? Why this either increase in the chromosome or decrease in the chromosome takes place? Just I have already told you in the last class. Okay, in a diploid organism, chromosomes are present in pairs, and this kind of pair of chromosomes are called as homologous chromosomes. Now, this is a normal. So instead, suppose if it is a 21st set, instead of 2 chromosomes, if 3 chromosomes are present, then this is nothing but a trisomy. We can call it as a trisomy. Okay, why there is an increase in the number of one particular chromosome? That is because of non-separation or non-disjunction of chromosomes during meiosis. During meiosis, you know that very well, chromosomes should get separated, but that is a failure of separation of chromosomes, and one chromosome it moves towards one pole, and which causes a disease like these chromosomal disorders. Now, very important is you must understand about the symptoms of this disease. Symptoms of disease. This disease will not be there for everybody, and it will not be transmitted by contact, because these are all genetic diseases. These are not contagious, and it should be carried from parents to their children. Now, how to understand? Out of 1,000, most probably out of 1,000 children, one children may be suffering from this kind of a disease. Okay. Now, what are the symptoms of this disease? How to identify? How to identify this kind of disorders when the children are suffering? Okay. Now, Down syndrome, mainly the individual affected is short stature. Short stature. Short stature means they are short with a rounded head. Round head. So this is what. So some scientists who observed this disease, they explain that almost a face and head is almost round like a full moon. Okay. So in this way, that stature is present, that character is present. Okay. Generally they are short and round head is present. And something slanting eyes are present. Slant eyes, which is not mentioned in your textbook. Slant eyes are present, which we can call it as a epicanthus. This condition is called as epicanthus. And one more thing is palm. We can understand from this also. Palm is broad. Palm is broad with palm crease. Palm crease are present. Palm crease is present. And physically they are very weak. Physical, okay, psychomotor development is less. Okay, psychomotor development is less. Still there are some more characteristics, and they have partially opened mouth, partially opened mouth, partially opened mouth, partially opened mouth is present, and because of that salivation takes place. Saliva will be okay, is coming out always. Okay, so these are all various characteristics by which we can understand about the children who are suffering from this Down syndrome. So now it is very, very important from the point of PUC in Karnataka. Okay, so sometimes they may ask to write this chromosomal complement, or which is also called as a karyotype, and another one is write the symptoms of a disease. So like this they will ask, and you should not forget to write, mention this one. Okay, physical psychomotor development is slow and partially open mouth with salivation. So these are all you have to write. So now this is one Down syndrome. Now let us move to one more chromosomal disorder, that is Klinefelter syndrome. So Klinefelter syndrome. Klinefelter's syndrome. These are all syndrome. We can call them as a syndrome only because it is a collection of diseases, collection of symptoms will be there. So therefore they are called as a syndrome. So this Klinefelter syndrome, so just I told you already, I am writing chromosomal complement or karyotype just in front of the name of this disease. Okay, so Klinefelter syndrome, so here it will have 47 chromosomes also. 47. In Down syndrome also 47. Down syndrome also they have 47 chromosomes. So where 45 in case of a Down syndrome, 45 plus XX, okay, is equals to 47. But here how it has become 47? Where the chromosome number has changed, where chromosome has increased. Okay, 47 with X, X, Y. Okay, in this way you can write, or otherwise still clear, to make it still clear, you can write 44A plus X, X, Y is equals to 47. This is very, very clear for the people. Okay, so here this 47, where it has become 47, this here in the 6 chromosomes. So it means there is a change in the chromosomes in the 6 chromosomes here. But from the point of examination, you have to write only this karyotype since it is there in the text. Okay, this is also right. Okay, which gives explanation for this. So it means here there is increase in the number of chromosomes and allosomes or sex chromosomes. Now, why this disease occurs? That is also, all these are all because of non-distinction only, non-separation of chromosomes during meiosis. One egg will receive extra chromosome and another egg will receive less chromosome, and the egg or ovum or gamete which receives more chromosome, if it is by chance fertilizer with the sperm, then it develops into this kind of individuals. So now, what are the symptoms? Symptoms of these diseases, disorders, Klinefelter syndrome. In case of Klinefelter syndrome, the very important symptom is overall masculine development. Overall masculine development. So here it affects the male. Okay, overall masculine development is one of the characteristic, and another one is feminine development. Feminine characteristics are developed. Feminine development means development of a female characteristics, particularly that is nothing but gynecomastia. Gynecomastia is developed in case of a male. Okay, gynecomastia is nothing but a development of a breast to some extent in male. So generally, which is a female characteristic, this female characteristic is developed in male. So when this is produced, male individual is sterile. And Here sperm formation, spermatogenesis, when male is sterile, spermatogenesis process which is taking place less, no spermatogenesis takes place here. Therefore male becomes sterile. So these are all few important features or symptoms of this Klinefelter's syndrome. In the examination they may ask to write about karyotype or symptoms of the disease. Then you have to list out all these symptoms regarding this Klinefelter syndrome. And one more, let us discuss about one more genetic disease, that is Turner syndrome. Turner's syndrome. Turner's. So here, as usual, I am writing this chromosomal complement here. Okay, in case of Turner syndrome, you can write 45. 45, just 45. With, what is the meaning of this 45? With X0 or O. X0 or O. Sometimes you may not understand what is this. Suppose if I write like this, it will be very clear for you people. Now 44 autosomes plus, okay, X0, it means this is 44 plus 1, therefore it is 45. Okay, now it is very clear, I think. Okay, now what is the meaning of this? Here, sex chromosomes or allosomes should be either XX or XY. But here it is not XX or it is not XY, 0 is there. Okay, so it means there is no one more X chromosome or one more Y chromosome is not there. Therefore, we have represented it as a 0. So 0 cannot be taken into account. Therefore, 44 plus 1 is equals to 45. Generally, which affects to the female individual. Now, this is karyotype. Karyotype of this syndrome or disorder, where this generally affects to the female, just I told you. Now this is also because of non-distinction of chromosomes and non-separation of the chromosomes. Now what are the symptoms here? What are the symptoms of this disease? You can easily understand that when the individual is suffering from this disease, they have, since it is a female individual, what characteristics she must have? Well-developed female characteristics or feminine characteristics like well-developed reproductive system and skin characteristic and voice characteristic. They are all exhibiting some female characteristics. So, but here that femaleness will not be there in a female individual because there is a rudimentary ovary. Rudimentary ovary is present. Ovary is a primary sex organ of a female. That ovary itself is not well developed. When ovary is not developed, no oogenesis, no menstrual cycle takes place, since because of this only one characteristic of female becomes sterile. Female is sterile. Why? Because she cannot produce egg, and another one is no menstrual cycle takes place, so that she is sterile only. Okay, so with this, we have discussed about different types of genetic diseases, or including gene disorders as well as chromosomal disorders. So thank you children. With this entire lesson of genetics, first lesson of your genetics unit is over. That is principles of inheritance and variation. And one more thing is here, from the point of examination, in the examination they will ask you 10 marks questions from Karnataka PUC. Okay, some of the important questions, one is monohybrid cross, generally they will ask, and another one is dihybrid cross, and another one is incomplete dominance. So these are very important for examination, and sometimes they may ask a question. They concentrate, question setters will concentrate on these genetic diseases also. And so these genetic diseases are very important from the point of CET as well as NEET. Okay, please prepare well and be thorough, and try to get the 10 marks from this lesson. Thank you.